it is exactly containing the heat kill mycobacterium W here. And in this heat kill mycobacterium W, it is having strong immunomodulator properties here. So that we are going to be discussing here. And this product has actually been available with this since almost like 20 years here. And with a long, number of clinical studies have been done in the, with the patients of tuberculosis, leprosy, anogenital wards. And last year, we got the approval to be used in the case of severe sepsis as well. So the molecule is well approved in the cases of, metastatic advanced non-small cell lung carcinoma cases, as well as the severe sepsis cases and some other, indications here. So, uh, but here we are going to be discussing about the, oncology part only rather than, talking upon the non-oncology, indications here. So shall I start?
Yes.
Yeah, perfect. So before going forward, I'll be just showing a, 3 to 4 minutes video clip here, which is actually made by the Kerala Pharmaceuticals to, tell you some introductory information about the Mycidac-C. So I'll be just playing this video, for you. I
The
hope this is
world we live in,
audible to you.
the air we
Yes.
breathe, the happiness we share. Imagine it all coming to an end. Imagine the cruel hands of a deadly killer snatching it all away. A killer named cancer. Cancer, the dreaded C word. Lung cancer, the most dreaded of them all. Lung cancer is the major cause of cancer-related deaths worldwide. More than 100,000 people are diagnosed with lung cancer every month. Much more than any other type of cancer. How does cancer develop? Immune evasion leads to cancer development and progression. Increased immunosuppressive mechanism leads to decreased immunosurveillance. Current therapy. All effective therapies improve immune function. Chemotherapy decreases immune suppression, increases immune activation, and is synergistic to immune therapy. Desmocolin 3, DSC3, is diagnostic of squamous non-small cell lung cancer. Chemotherapy converts DSC3 negative to DSC3 positive cells. It is also expressed in other cancers like melanoma, colorectal cancer, besides many others. The emerging treatment, Mycidac-C. Mycidac-C is active immune therapy targeting DSC3 expressing cancer. Mycidac-C activates immune cells and expresses DSC3 on their surface. Mycidac-C reduces immunosuppression by its effect on immunosuppressive cells and cytokines. It preferentially kills DSC3 expressing cancer cells. Mycidac-C provides synergy to chemotherapy. All this backed by solid research and innovation. Randomized controlled clinical trials have been conducted wherein Mycidac-C is added to standard chemotherapy to evaluate its safety and efficacy. Research studies have proven that addition of Mycidac-C does not add to toxicity. Addition of Mycidac-C improves response rate. Addition of Mycidac-C improves overall survival probability by 28.2%. Improvement in survival in squamous non-small cell lung cancer (NSCLC) is 43.5%. This is significantly better than most other therapies. The future. Mycidac-C, a game changer in the treatment of lung cancer. Cadila Pharmaceuticals' extensive research relating to the treatment of lung cancer has brought a smile to many a face. All this would not be possible but for the vision, persistence and foresight of Shri I.A. Modi. His thinking was very clear. Anything that can add to a moment of life and make it more meaningful is definitely worth the effort. Mycidac-C offers that hope for life.
Thanks for seeing this movie. Let us come to the presentation here. So before, I hope this is visible to you.
Yes.
Perfect. So before going forward, I would also like to introduce my our R&D head, Dr. Baklesh Kumar. He's also available with us today. Kumar sir, hello. Kumar sir, are you able to hear me?
Yes, yes. Good
Yeah.
evening, everybody.
So our R&D head, Dr. Kumar, is also here. Dr. Deepak.
Yes.
Yeah. So let us forward. Let us move forward here. So this is regarding the immunotherapy in the cases of in the cases of in cancer. So this is regarding the history of immunotherapy. Here, like when we talk about the chemotherapy usage of the chemotherapy in the field of oncology, it started, somewhere around like 1942 with the discovery of the nitrogen mustards, and over a period of time we used methotrexate 1958, and followed by the vincristine, doxorubicin, tamoxifen, and now at the moment we are using the tyrosine kinase inhibitors and the other chemotherapeutic agents in the oncology. And when I talk about the immunotherapy, the story is not too long here. It started with the discovery of the interferon alpha in 1986 only, and followed by the usage of the BCG, like in the case of superficial beta carcinoma, we are using it, and followed by the monoclonal antibodies like the trastuzumab, bevacizumab, many others. And talking about the Mycidac-C has been shown here, like we got the approval in 2013 to be used in the oncology from Drug Control General of India, and it is approved for the advanced non-small cell lung carcinoma cases, which I'm going to be elaborating here. So this has actually been, published in Times magazine, like how to cure the cancer. So there was an article being published, in this Time magazine, like which is actually been talking about the immunotherapy to be used in the oncology field. Now talking about this further here. So this is regarding the immune mechanisms in the oncology. Like there is a escape phenomena is there, like T cells are there, but they're not able to recognize the antigenic surfaces upon the tumor cells, and that is why there is a killing is or the cytotoxic effect is not there in the development of the tumor. So whatever people are doing with the immunotherapy, we are trying to build up the immunity, increasing or activating the T cells, increasing the number so that these T cells they're going to be recognizing them, and there is going to be the enhanced cytotoxic effect. Same is being done by the our product Mycidac-C here, which I'm going to be elaborating here. Now, this is being done in the laboratories of the Ketla Pharmaceuticals, like which is trying to tell the effect of Mycidac-C immunization upon the effect of the T cells. Like if you see like left picture here, like it says like whenever there is going to be multiple injections of Mycidac-C are to be given, it are going to be given to the person who is suffering from the non-small cell lung carcinoma. In these cases, Mycidac-C is going to be inducing the expression of the desmogality on the surface of the T cells. So it is exactly been correlated with the number of injections here. More number of injections are going to be there, and ultimately there is going to be the enhanced expression will be there upon the T cells. And right-sided picture it shows like the killing is going to be much more in the desmocolin 3 positive cells, which are characteristically found in the case of squamous variety. There is a more level is there of desmocolin 3 on the surface of this squamous variety of the non-small cell lung carcinoma, and same is being correlated with the cytotoxic effect. More is going to be injections of Mycidac-C are going to be used and simultaneously correlated with the cytotoxicity. Now talking about this further here, like as stated, like uh, giving the exposure of the chemotherapeutic agents, so even in the subtherapeutic doses, it is going to be creating the desmocolin 3 on the surfaces of the T cells. So, like after giving the chemotherapeutic agents, it is going to be creating the desmocolin 3 on the surfaces of the tumor cells, has been shown here. But after giving the injections of Mycidac-C, it is going to be producing the desmocolin 3 on the surfaces of the T cells. So there is going to be the enhanced killing which is being correlated here. So this is regarding the chemotherapy and the effect of of production of the or development of the desmocolin on the surfaces of the tumor cells. Now talking about this further here, this is the mode of action of Mycidac-C. Like it induces the expression of the desmocolin 3 on the surface of the immune cells, and desmocolin 3 expressing immune cells they to kill the desmocolin 3 expressing cancer cells. And the statement is there like desmocolin 3 is the marker for the squamous variety of the non-small cell lung carcinoma, which again I'm going to be elaborating in my further slides. Further, desmocolin 3 negative cells, they do express the desmocolin 3 following the chemotherapy, even in the subtherapeutic dose, which I've shown you in my previous slide. Now, this is regarding the details of Mycidac-C injection. Like as I said, we are the innovator for the molecule, and it was developed by the Kedla Pharmaceuticals, and it is an immunomodulator. So it increases the immunity of the person where the injection is going to be given, and the components of zero point 0.1 ml it contains like heat kill micro W, as I said, and the number of bacilli here are zero point 0.5 multiplied with the 10 to the power 9 here, and each injection is available as a pre-filled syringe here, and where the content is zero point 0.2 ml here. And talking about the excipient here is the sodium chloride, and talking about the preservative agent, it is containing thiomersal here in a very low quantity, so no risk of toxicity is there. Now, talking upon this further, like this is regarding the site of administration where the injection is approved to be for the administration. Like here, the four sites are selected like over the deltoid region. Second is upon the suprascapular region. Third one is periumbilical region around the umbilicus, and even on the inner part of the thigh, the injection is available. So here, the route of administration is intradermal here. So here are the sites where injection can be given easily. Now, talking about this, this is actually talking upon the immunohistochemistry using the commercially available stain, which is talking upon the presence of desmocolin 3 on the surfaces of the tumor cells. Now, this is what I was trying to tell you here. Like this is talking upon the literature, which states that desmocolin 3 expression is more on the surface of the squamous variety of the non-small cell lung carcinoma. So, if we talk about the squamous variety here, like what et al. Shows like the 93.2% of desmocolin 3 expression will be there, and same is correlated with the other other published literature like 91%, 71%. So when we combined all of this data, which comes out to be around 86% of the these tumors, they are going to be expressing the desmocolin 3, which is just exactly opposite to that of adenocarcinoma, where the desmocolin 3 expression is almost minimal, like 0.45% we got it. And further talking about the other varieties like the last cell, again the expression is there like 11.2%. And this what we are trying to tell like desmocolin 3 is going to be is being the marker for the non-squamous variety of the non-small cell lung carcinoma. Now, from here I'm going to be elaborating upon the clinical studies which have been done upon the Mycidac-C. Here I'm going to be talking upon the initial pilot clinical study. followed by the approval clinical trial of Mycidac-C in the patients of advanced non-small cell lung carcinoma. So this is the first clinical study which has been done with the Mycidac-C. So it is a desmocollin-3 open label randomized controlled clinical trial of desmocollin-3 targeted toll-like receptor agonist Mycidac-C in the patients of advanced non-small cell lung carcinoma. So post-randomization, like in the study arm, the Mycidac-C intradermally was used with the doses of 0.1 ml every fortnightly up till 6 months. And talking about the chemotherapies, two chemotherapeutics are selected here, like cisplatin with the dose of 75 milligram per square meter of body surface area plus etoposide with a dose of 100 milligram per square meter body surface area. And upon the control, both of these chemotherapeutics they were used here, and the patient continued till the 6 chemotherapeutic cycles. Now, this is regarding the patient characteristics here. Uh, when I talk about the patient characteristics in the terms of age, sex, Karnofsky score, stage of the disorder, both of the arms they were similar in nature. Now, this was the output of the clinical study. Uh, when I talk about the response on the patients, like it was 11% better in comparison to the control arm. So in the Mycidac-C arm, it was 38% versus the control arm where it was 27%. And talking about the progressive disorder, like a lesser number of the patients, they do progress after receiving the injection. They were 19% in the study arm, but in the control arm, there were more number of patients who actually progressed with the disorder. They were 27%. And one patient who actually got the all reversal or suppression of the symptoms. So it was a case of complete response, which was seen in the study arm but not in the control arm. So this is regarding the advanced technicians which were being uh, you know studied here. So in terms of the hematological as well as the non-hematological side effect, both arms were equal or rather. In the Mycidac-C arm, there were lesser number of the adverse reactions, lesser number of side effects they were being found here. So, so this is regarding the approval clinical trial of Mycidac-C. So this was the open level randomized controlled clinical trial of desmocollin-3 targeting toll-like receptor agonist Mycidac-C micro-W in the combination with platinum-containing doublet paclitaxel plus cisplatin versus the only cisplatin plus paclitaxel in the advanced non-small cell lung carcinoma cases. So post-randomization, the cisplatin with a dose of 100 milligram per square meter body surface area plus paclitaxel with a dose of 175 milligram per square meter body surface area, and the cycle duration was of 21 days here. So immunotherapy which was used here was Mycidac-C intradermally, and it started with the 7 days prior to the chemotherapy, like 0.2 ml, 0.1 ml upon the one dental teacher intradermally and 0.2 ml, 0.1 ml upon the other arm. So after giving the chemotherapy on the very first day, on the 8th day, the another injection of 0.1 ml was given, and on the 15th day, another 0.1 ml of the injection was used. And for the subsequent chemotherapeutic cycles, these two injections were continued. So zero point 0.1 ml on the 8th and zero point 0.1 ml on the 15th day. So 9 injections were given during the time of the chemotherapy. And post these four cycles of the chemotherapy, the injection was continued here. So this is how the statistical analysis were done in both of the arms. So overall survival was noted, and the safety parameters like the adverse conditions they were being evaluated here. So this was the eligibility, as I said, like the patients were actually being suffered from the metastatic uh, advanced non-small cell lung carcinoma. Age was 18 or 18 plus, and the echo criteria was 0 to 1, and the life expectancy was 24 weeks or more than that, and the neutrophil count was 1500 or more than that, so that they can tolerate the chemotherapeutics, and similarly was correlated with the platelet count and hemoglobin levels 9 or 9 plus. Now this is regarding the eligibility. Further eligibility like liver functions were within the range, and the renal function like same criteria was lesser than 1.5 or equal to that, and the pregnancy was excluded or ruled out here, and the informed consent was taken out. Now, this is regarding the demographic characteristics in both the arms. So, as you see here, like in both of the arms, they were similar in the terms of age, gender, ECOG criteria, and stage of the disorder, stage 3B and the stage 4, they were recruited for the study. And talking about the histology, like adenocarcinoma cases, squamous, large cell, other types, they were included for the study. Now, talking about this output of the clinical study, uh, when I talk about the overall survival, like which is comprised of complete plus partial. So in the Mycidac-C arm, there was overall 10% advantage was seen in versus the control arm. So in the control arm, like where the platinum containing doublet were used, there was 37%. But in the case of study arm with Mycidac-C, it was 47%. Talking upon the progression of disorder, there were 11% with the study arm. There were lesser number of patients, like 5% lesser in comparison to the control arm. There were 16% of cases they were actually progressed with the disorder. And the another good part of the a good part among, from the output, there were three cases of the complete response. They were seen in the study arm, which was containing the immunomodulator Mycidac-C, but none in the control arm. So it was concluded that addition of the Mycidac-C is definitely going to be improving the response rate in such patients. And talking upon this further here, like this is regarding the progression-free survival in the patients who actually complete the 4 cycles of the chemotherapy. So the arm where the Mycidac-C was given, there was the progression-free survival of 253 days versus the study arm, there was a progression-free survival of 157 days. So it was almost 97 days better in comparison to the control arm. So hazard ratio was pretty beautiful here, like 0.43 we got it post study. Now, this is regarding the overall survival for the patients who complete the 4 cycles of the chemotherapy. So, with the study arm, which was containing the Mycidac-C, here it was 299 days versus the control arm, there was 233 days. So, overall 66 days advantage was seen after the use of our immunomodulator Mycidac-C, and the hazard ratio here it was 0.64 here. Now, this is regarding the best outcome of this clinical study, like in the terms of overall survival for the patients who actually been suffering from the squamous cell variety of the non-small cell lung carcinoma. So here the advantage of overall advantage of 100 days was seen versus the control arm. There it was 254 days, but in the study arm here it was 364 days. So this was the best output we got it after the previous study. This was 0.40 of hazard ratio. We got it after in the cases of squamous variety. Now, this is regarding the adverse reaction. So, uh, when I talk about the hematological side effects in both of the arms, they were equal or they were better with the Mycidac-C here. So, talking about like anemia, leukopenia, or neutropenia, both of the arms they were almost equal here, or rather they were better with the Mycidac-C. So, this is regarding the uh, non-hematological side effects which were noticed after the clinical study. So in the terms of dyspnea, diarrhea, nausea and vomiting, and fatigue, both of the arms, they were almost equal here. So it was concluded that addition of the immunomodulator agent Mycidac-C, it is not going to be increasing the side effect, whether it's going to be hematological or whether it is going to be non-hematological. So these side effects, they were not being seen here. But some cases of the dermatological, injection site reactions were seen in the arm where the Mycidac-C injection was used. Now, to conclude here, like Mycidac-C is synergistic to the chemotherapeutic agents. It was concluded here, and definitely it is going to be improving the progression-free survival with a hazard ratio of 0.43, and it improves the overall survival in such patients. And all over it was hazard ratio of 0.64, and the bus output, as I discussed, here it was 0.40 with the squamous variety of the non-small cell lung carcinoma. And actually it is not been associated or it was not found to be having any kind of the systemic side effects, whether hematological, non-hematological. And this was true with the patients who actually complete the 4 cycles of the chemotherapy. Now, all this data has already been published by, one of the, best oncologists from the globe. He is none other than Dr. C.P. Chandra Benani, and our R&D head. He also, contributed. He, got the paper published here. So this been published in the Annals of Oncology. So after this, session, definitely I'm going to be sharing this, paper with you so that you can, go further upon that. Now, talking about this further, like the other varieties of the other sites where the desmocollin 3 expression is there, like in the case of urinary bladder carcinoma. So this clinical study upon such patients is already been done. So it was concluded out like in the case of superficial bladder carcinoma, we are going to be giving the intravesical BCG administration. So similar results oncologists they're getting after the usage of the intradouble Mycidac-C here. So this is under the process of approval from the drug control general of India. So as soon as it is going to be having the approval, definitely we are going to be communicating this information with the oncologist, and it is going to be under the active promotion. So that is why for this indication we are not doing the active promotion, but it is under the process. Same is true with the ovarian carcinoma, and same is true with the colorectal carcinoma. And talking about the melanoma, some studies are already been done here, and same is true with the meningioma, and for the head and neck tumors, some studies are already been done. So if you permit me another 5 to 7 minutes, definitely I can share the data as well with you. So this is regarding the that earlier clinical study which has already been presented in ESCO in 2011. So this is open label randomized multicentric phase 2 clinical trial of toll-like receptor Mycidac-C KD05. So earlier brand name of Mycidac-C was KD05. So it has already been presented in ESCO. So it states that median overall survival was 236 days in the control arm. It was improved to 59 days to 295 days in the KD05 arm. And there was no increase in the adverse degradation. So this is another presentation which was, done. So by Dr. Chandra Birani. It states like addition of the KD05 to the paclitaxel plus cisplatin in the patient with the advanced non-small cell lung carcinoma is safe and results in the improvement of progression-free survival as well as the overall survival. So this is another one. So this is a recent publication we got it in the case of the head and neck tumors. So it states like the title is a comparative evaluation of Mycidac-C based immunotherapy plus concurrent chemo radiation versus the concomitant radiation therapy in the locally advanced head and neck tumors. So it concluded that like this study on the Indian locally advanced head and neck squamous cell carcinoma patient says that the usage of the weekly micro W vaccine plus weekly cisplatin is safe, and micro W with concurrent chemo radiation therapy may be superior to the radiation therapy or chemo radiation therapy alone in the terms of the better response rate, higher local regional disease control, and with the lesser toxicity. So this is another publication which had been done by Dr. M.C. Pant from Lucknow. So this is another work has been done about the head and neck tumors. So which conclude like immunotherapy may be useful as an adjuvant therapy in the head and neck tumors. Receiving the chemo radiation for the better clinical response, reduction of the side effects, and improvement in the quality of the life. So that's it from my side. Yeah, Dr. Deepak, any queries here? So as I like elaborated, like we are under the process of doing the more clinical studies here. So some oncologists they are like as I told you during the time of clinical study, like some cases of the injection site reactions were there. So at the moment, like people are using it by intravenous way as well, by the pre-medication of the dexamethasone, since it is a immunomodulator here. So they're also using it with the intravenous, injections here. So in Tata, like under the leadership of Dr. Kumar, was some IV, the clinical studies being done, and there's another clinical study which is being under the process by at Bikaner uh, under the leadership of Dr. Surinder Peniwal. So, we are trying to explore the indications here, and it is under the process of usage here.
So my question is, this is not a very new molecule. It is at least 15
Yeah,
years since
no,
it is available.
talking about the oncology, we got the approval 2013 only, so it is 7 years old model.
Okay, 7 years old.
Amit,
And
Amit,
and some
don't argue. Please listen. Listen to him.
Sure, sir.
and some of these studies, they have shown a OS advantage, overall survival advantage.
Yeah, overall survival advantage is the progression-free survival.
Yes. So normally what happens that in lung cancer or any other cancer, if a drug is showing an overall survival advantage, that is by about 2 months or something, in one study we have shown 3 months also.
Yeah.
So then that drug becomes a means blockbuster. It becomes a super hit. So why in last 7 years it has not happened with this molecule?
It is because people in many number of cases they wanted to have the product into the guidelines. Like they complete like your product is not being mentioned in the NCCN guidelines, so they're finding it difficult to during that point time to to judge the product or to use the product. So we are under the process like uh, I would say rather people who have used the product there are very few here. So for this
Well,
reason.
Dr. Deepak, this is Rajiv Saxena. Good evening. Greetings.
Good evening.
How are you?
Fine.
Happy to have you. Right. And very, very apt and pertinent question, sir. And then as soon as I took over charge here in Kerala after working with Zydus for 10 years in oncology and before Roche and Intas, sir, what has happened is, we have not actually taken up this drug on the serious platform as a company, as a marketeer, and reaching out to medical oncologists talking about it. So perhaps it is lacuna on the marketing side when we need to reach out to the medical oncologists, understand from them that how the product is behaving. At the point in time when this product got launched in India, there was no Opdita, there was no Keytruda was available. And then the extension of line therapy in the lung cancer was the most awaited. And then this product when launched was very welcomed by the medical oncologist. However, the instability of the team who are going out and talking to medical oncologists, seek their opinion on it and keep on doing some, phase 4 studies, which will help us understand how it is behaving further. This is one. Second is, of course, yes, sir, one point in that in medical oncology, most of the product been taken up, which is FDA guidelines available or NCCN guidelines available, which has been take up very easily. However, it is Indian innovation. And to get into that, therefore, Dr. Chandra Bilani was there, who was working on this very product. And we wanted to get into the US and then get into the various guidelines possible this product, looking at the benefits shown by the product itself in this condition. However, it has been used in several other conditions, sir. One such is sepsis. It is the only drug approved in India for sepsis, sir, today. And then there are a lot of doctors who are using it. Dr. Randeep Guleria himself talks about a lot on this very product today. It has been also tested in COVID patients. It's going and a lot of benefits coming out because it works on IL-6 pathways as well. So it actually stops the cytokine storm. Now, when get into the US clinical trial, it is required 10 billion US dollars, sir, for to be very precise, get into the trial approval and get into US, and then there's the lobbying any drug in India. There are a lot of drugs being manufactured by Biocon, but still, difficult to get. But the Biocon manufactures trastuzumab, it easily gets approval as biosimilar there in Europe and India. So that's where, sir, this is thing, and we are looking forward to we start establishing, first of all, in Indian subcontinent, we can manufacture, market this drug, acceptance, medical oncologists like you, start accepting, okay, there is something available in this condition. So when I compare Opdita or Keytruda, there one single cycle therapy is costing around 2.5 lakh rupees currently. Whereas Mycidac-C, what we are projecting is the Indian immunomodulator now, which is actually available around 5,000 rupees, 4,900 rupees MRP, pre-filled syringes. The entire treatment cost is around 45 to 50,000 rupees already, only 4 to 6 cycles been given. So we are just debating on that how to go, and therefore these meetings are now we are planning, sir, reaching out to doctors like you, like Dr. Kumar Prabhash is also now working on it. He's already using in Tata, approximately 100 to 200 PFS we are selling there, approximately 15 to 20 patients regularly been put up on there. Dr. Somani in Bhagwan Mahavir Cancer Hospital is starting using. Dr. Apurva Patel in GCRI. Dr. Prem Kumar Devstos in the in TNMSC. It is approved TNMSC also. It is approved in TMH Bombay in the protocol TNMSC. RCC here in Rajkot, Ahmedabad, GCRI. So we starting now. It's also approved now in Punjab Health Government Services, and we got it approved now into the Bikaner Medical College, Acharya Tulsi Medical College also. So we started working on, and we lagged upon reaching out to the doctors like you, medical oncologists, wherein the acceptance come and we learn from you how to go forward, understanding this product behavior, how it's behaving. So I think that's where we missed out. One is, of course, yes, NCCN guideline is something which is very well welcomed by medical oncologists. However, it is an Indian innovation, Indian product. We are working on it. How soon we get into, and then Dr. Khamar himself is here, who has invented this product. Sir. He joins us. So, Dr. Khamar, welcome. If you would like to add something beyond what I said, and then correct me.
So, okay. So, prima facie, as you said, this product is a long drawn history, and it took little more time than the average time because we had to start from scratch. If you remember, when we started developing this product, immunotherapy was not known in cancer as a successful story. So it took us a lot of time to understand. So we did a lot of basic science work internally to understand what is it doing and how it is doing. And luckily, we could identify the biomarker, which is rare for this kind of a product. So we could identify a biomarker and show it that this is a biomarker, and wherever it is present, it works. Luckily for us, this biomarker was also being used because of the Avastin for differentiating non-small cell lung cancer, squamous versus non-squamous. So it became very handy because a lot of literature was available, and luckily that was the biomarker for us. So that helped us in moving forward. And then we took it forward as an intradermal just to pass through very quickly. But we parallelly did a lot of experiments to see how we can change the route of administration. So, this product can be given by any route of administration. Only thing is you have to change the dosage and you have to adjust appropriately. So when you give intradermal, I mean, there is a local side reaction, no other side reaction. But when you give IV, it has, like any biological, it induces infusion reaction or transfusion reaction. So to take care of that, we recommend that you give 2 cc of dexamethasone before you give this, but the efficacy is maintained, and we have generated animal data for that. So we are, trying to make it more user friendly because it is working. We are trying to make it more user friendly, and just to tell you, one of the animal study long back was done in USA. And when they did the animal studies in the melanoma, that model is a very aggressive melanoma model where they inject large amount intravenously so that they metastasize in the lung. And according to the group, even today, for that model, there is no effective therapy other than Mycidac-C. When they used Mycidac-C, they found that the metastasis to the lung was delayed and there was improvement in survival by 33%. So typically all animals will die by the end of 2 weeks. This animal will survive for 3 weeks. And then the metastasis is delayed. So at early time point, there is no metastasis to the lung. But subsequently, you see the difference in the sizes because it's so aggressive model where no product has worked so far. So based on that, the same group did a clinical study in US in melanoma, and they showed that as a monotherapy, it is possible to get no evidence of disease in melanoma, but not in all. And there they use a very high dose. Instead of giving every fortnight or every week with the cycle, they gave it twice a week for 8 weeks, and then they evaluated. And then those who got some success, some response, they were again given for a 2-month cycle. And like that, they continued. And then they showed that even in advanced melanoma, where there was no treatment at that point in time, this showed some efficacy in terms of no evidence of disease. Okay, so this was a small trial done. But then as it happened, the immunotherapy started becoming And then people started losing interest in products from a small company with an active immunotherapy. So far, if you look at it, all the drug which has worked from immunotherapy perspective are passive immunotherapy. That means you do something to remove the immunosuppression. The beauty of this product is while it is active immunotherapy, it also takes care of some amount of immunosuppression. Tregs are reduced with this product. And that's the reason it works as a monotherapy in melanoma. It worked as a monotherapy. And the guy who did is, luckily, at that point in time, he was an oncosurgeon with PhD in immunology. So he did a lot of work and he helped us understanding better. So in this background, if you look and compare with other therapies, since it's active immunotherapy, there are lesser side effects. So only side effect we have seen is if you give intradermal is a local site reaction, but if you give IV is a transfusion reaction, and which is not there if you give dexamethasone as a therapy prior to it. Now, this is the, and then because it is active, it does not work on its own. It boosts the immune response of your body. So when you give a small amount, zero point 0.1 or zero point 0.2 ml, it changes the immune profile of your body. So that MW or Mycidac-C, which is given intradermally, is taken to the neighboring lymph node, and there it processes like any vaccine. But when you give a vaccine, it produces antibody. When you give Mycidac-C, it produces immune cells, activated immune cells, which expresses desmocollin 3. So it works by cell-mediated immune response, which is a novel concept. See, when Keytruda or nivolumab came, they were not knowing how it works, but now we know that it is a proliferation of antigen-exposed immune cells within the tumor, and that's how it works. So this sensitizes body's own immune mechanism, and it works. So that's the reason probably there are minimal side effects or no side effects. And I can go on and on, but I would like your feedback and comments so that I can provide you more relevant information. I can go on because we have worked for so long on this product. I have tons and tons of information.
So, I have, for the first time, I have heard about this product, that there are phase 2 studies, phase 3 studies, which have shown higher response rate, higher progression-free survival, overall survival benefit of about 2 months. So what happens it's normally what happens in any cancer, like in pancreatic cancer, addition of erlotinib to the chemotherapy, they had a 15 days increase in the survival, and that became the standard of care at that time. And 15 days was also very important in pancreatic cancer. And in lung cancer, most of the studies, they have shown all the molecules which have been approved survival difference of about 2 months.
Yeah, maximum.
Absolutely, sir. Even cetuximab,
Okay,
bevacizumab,
Yes,
regorafenib, all them.
Okay. So he has opened an interesting concept. So what was not shown in the slide is you touched upon pancreatic cancer. So we analyzed pancreatic cancer samples, TMA, provided by Stanford University, and we realized that though it is not publicly known and not evaluated, pancreatic cancer is desmocollin 3 positive. And gemcitabine and Mycidac-C has a significant synergy. We have done animal studies. We have done pancreatic cancer model animal studies, and there's a tremendous synergy. So in that model, I'm just digressing, but providing you some more information so that you can look at it and then come back to us that what we should be doing. So that animal studies, we demonstrated that intratumoral active T cells increases when you combine gemcitabine with Mycidac-C. There is a decrease in PD-1 cells or immunosuppressive cells intratumoral, Treg intracellular, and there's a tremendous synergy. Currently I don't have it, but I can share that at appropriate time, that information which we have done in animal studies, and that prompted us to look at whether desmocollin 3 is present in actual human samples, and we found that it is also present. More than that, this is not known to all of you, okay, which we are doing. So we are doing some bioinformatics. So we are looking at TCGA data set and trying to understand what is right, what is wrong, and what is the relationship. So hopefully in next 3-4 months, I will provide more information or publication on that. So we have done some data set. So what is happening and why it is happening that way? So I mean, from a comparison with, I mean, so there are a couple of things. So I mean, just if I stick to the lung cancer part, so in the lung cancer, squamous cell carcinoma is a desmocolin 3 positive majority. So that's the reason we are getting more response there, which is 3 months advantage. If you look at adenocarcinoma, now currently adenocarcinoma is either chemotherapy or targeted therapy. Now, when you give targeted therapy, and this is important, Rajiv, for you also, this may be news. When
Yeah.
you give targeted therapy, with a targeted therapy, checkpoint inhibitors don't do well because targeted therapies are associated with immune activation and there is no suppression. So, wherever targeted therapies are added to a checkpoint inhibitor or vice versa, there is no synergy seen. But with chemotherapy, uh, immune checkpoint inhibitor, there is a synergy seen. Now, I mean, if you go back to the basic, when you give any therapy which provides response or likely to provide response, in adenocarcinoma, the desmocolin 3, which was not expressed, starts expressing. And another group has already demonstrated and proven and published it. Okay, when you have EGFR inhibitor, desmocolin 3 comes on the surface of adenocarcinoma. And then there's a minimal immunosuppression, so the checkpoint inhibitor don't work. Now, if you take it this up and add Mycidac-C, then there will be tremendous synergy, and hopefully the response rate will increase, but more than the increase in response rate, the durability of the response will significantly increase. So this is our, I mean, working hypothesis. We have not actually done this. Okay. So this is something which is, we have realized that because of the checkpoint inhibitor not working well, we dug out the information and came to this conclusion, and then we worked around it. So this is for a lung cancer. So targeted therapy, non-targeted adenocarcinoma, simple chemotherapy, and this. Now the other part is, as I mean, this is a relatively recent, as per desmocolin 3 expression is higher, and this is based on the bioinformatics. So at a gene level, you can always identify that desmocolin 3 is higher and desmocolin 3 is lower or desmocolin 3 is absent. So when the desmocolin 3 levels are high, that tumor is more likely to have a lesser immunosuppression compared to higher immunosuppression. So we have seen this in bladder cancer, for which we are writing a manuscript. So in a bladder cancer, which Amit touched upon, so we have a publication where we have shown that BCG refractory bladder cancer or BCG recurrent bladder cancer, MW given intradermally has a response, and that response is durable because, and this was done under US FDA. See, all these studies are done under IND with US FDA, but we have not been able to move forward because of the lack of funds and tremendous competition coming from the passive immunotherapy. Because by the time we reached there, nivolumab started coming up, CTLA4 ipilimumab was already in the pipe, there approved, and this started coming back. So we took a back seat because we can't fight with those giants. We don't have those muscle power and all, and then we need some backup. So that's the reason we put it on a hold. I mean, this is a real that we put it on a hold deliberately, that okay, let's wait and see where do they go and how far they can reach and what are the vacuums created by them. So I mean, from Indian perspective, vacuum created is, first is a cost, and second is, I mean, when we analyze the data in a squamous cell, this is still the best. Even if you don't use them, just use chemotherapy plus MW versus chemotherapy plus say pembrolizumab or pembrolizumab, and those the response rate, and the recent one is supplement. So response rate and the overall survival benefit is still better with Mycidac-C plus chemotherapy. So now we feel a little more energetic to take it forward, because now whatever has to happen with checkpoint inhibitor has happened.
Yes.
They have reached their plateau now. Okay, they were moving up, but now they have reached plateau, and you don't see a lot of noise coming. So for us, this is the right time to activate our program and seek your suggestions, blessings, guidance, because now you are more inclined for immunotherapy. When I started, everybody was laughing at me that what are you doing? You are wasting your time. I mean, as a matter of fact, the study which you saw, the data, it was approved by US FDA, and we presented them. So their first question was, do you have extra money? Give it to us. Why you are wasting your money and resources? You are a small company. This was a feedback from US FDA. Okay. So under that circumstances, we have reached where we have reached.
And sir, we also do the estimation of DSC3.
Yeah,
There
yeah.
are multiple patients, we get the sample done,
No,
and
but
then
it
we
is
get the
easy.
DSC3
Okay.
estimations as
I
well.
mean, gasmocolin 3, if you want to establish at your place, it is a simple immunohistochemistry, and the kit is, antibody is readily available. And that was the advantage we had because somebody had worked on it because of the bevacizumab, and there were publications on it, so we could jump on it. So it's not a difficult. Okay, I have spoken enough. Now I have to listen to you.
My question is very simple. I that any drug which shows a survival difference, like this molecule has shown a survival difference, which is very, I will say that out of 100 trials,
Yeah.
2 or 3 will show a survival difference, and that becomes the standard of care.
Yeah.
So it is nearly 7 years, and means
Yeah.
I have never heard about this molecule.
Yeah.
There is a publication of Paclitaxel versus Paclitaxel with this molecule. Why it has not come into the eyes of the entire world?
Yeah.
This is
So
my question.
yeah, that is a Raj's job, not my job. Okay.
Sir, as I and
Because
you are
this 2 months OS difference becomes an NEJM article, NEJM publication, like Paclitaxel with bevacizumab. That was a NEJM article of 2 months difference in survival.
absolutely,
So,
sir.
so there is something lacking, or from your part, that
Yeah.
this
Yeah.
molecule
So
has
In
not
my
become a.
okay, hold on. I
if
mean, just
I
I'll tell you. So when we sent an application, I mean, it was immediately accepted as an oral presentation with a discussant in ASCO and World Lung Congress. So it speaks, correct? Now, at that point in time, immunotherapy was in offing. And the challenge which we had was to convince people that chemotherapy and immunotherapy can coexist and they can be synergistic. So that was a big hurdle we had. Okay, because the thinking was different at that point in time. Luckily for us, checkpoint inhibitors did all the spade work, which we could not do. We don't have a muscle power, we don't have that bandwidth, and so everybody looked at it with a suspicion. Okay, and so we said, okay, what do we do next? So Dr. Bilani says, okay, don't try for a high fly journal, go for a journal which is middle level. Okay, and that's how we went to Annals of Oncology, and with the first instance it got accepted. There were few comments which we had to respond, but we did not find rejection. हमने भेजा, accept करके वापस आ गया. Okay, and that was a strategy we planned, and hopefully, I mean, it turns out that we were successful. Now if you look at it, there was a time gap because we waited. So if you look at a publication, this happened only in 16. Publication happened in 16, presentation was 11. So we allowed that time gap for things to mature. And when it matured, we could get publication easily. But before that, I mean, whenever I was myself present at ASCO for oral presentation at World Lung Congress, and everybody was looking at this with suspicion. How can it happen? How can it happen? How can it happen? Because there was a background knowledge was deficient for an average oncologist. So, I mean, I was advised by immuno-oncologists of Johns Hopkins who were behind the checkpoint inhibitors. I can name a few. And all those were very gaga after me. But then it stopped because I can't take it to the US. Because in US, we don't have a, I mean, system where somebody else will pay it and I don't have to pay. So they are still very, I mean, they always used to say, tell me. I was interacting with them for more than 5 years, and they will always tell us, you have a product and we have a science. Your product works, our science does not work. And they are behind checkpoint inhibitors. But luckily, things have changed. So if Indian oncologist starts using it, generating data, and creating some goodwill about the product, it can again go back and start coming back again. Because now, if checkpoint inhibitors have done whatever they wanted to do, they have educated people, and they have also shown what it can do, what it cannot do. Like one of the queries was, in immunotherapy, you see a tail effect, correct? So which was not known at that point in time. So everybody says this is not possible. How do you get a tail effect? Then there was another challenge is why chemotherapy has to be 4 cycle complete. And what happens if they receive only 1 cycle? Because chemotherapy also helps in reducing immunosuppression. That concept was not there at that point in time. We knew it, but that concept was not well established. So why we have to have, when you have a large size tumor, there's a tremendous immunosuppression, and chemotherapy helps in getting rid of immunosuppression, reducing the size of the tumor. And that point in time, if you add immunotherapy, which is active immunotherapy, it will help. And luckily, some of you now, I mean, I'm told, because I'm no more active with talking to oncologists, that as a monotherapy, like we showed in the melanoma, somebody in India has shown it as a monotherapy in advanced lung cancer. It has been successfully. Correct, Rajiv? Am I right?
Yeah, true, sir. True, sir. Very
True,
true.
sir. We recently did this work. Dr. Amit would like
Yeah,
to.
So
I'll
what
elaborate
I'm
here.
saying is more and more people start thinking around it, using around it, they will generate users experience, and this will be widened. See, we are not even going beyond lung cancer currently. As I mentioned, pancreatic cancer is a good candidate, but we are not moving forward because we want to consolidate. You generate data, we will provide you all the support, but you generate your feedback, your analysis, what it is does, what it does not. We have done the science part of it.
Very recent developments are this. We have done the lung cancer monotherapy Mycidac-C. Dr. Amit have received the all data.
Yeah. So at the moment is under the process of publication here. So it is done by
But
the,
you can describe. You can
yeah,
describe
Please
what
definitely.
was the
describe. Please
patient,
describe.
So
under what circumstances it
actually,
was given,
this was the
how it was
60,
given.
yeah. So this was the actually the 65 years old case who was actually being suffering from the squamous variety of the non-small cell lung carcinoma. And this person was actually in the very bad
You have an X-rays or
condition.
CT scan. I mean,
Yeah,
we
that I do
appreciate.
have it. Shall I
That was
show
good.
it to you at the moment?
Yeah.
Yeah, sure. Just give me a second.
See, he is a real person, so he will appreciate. I am not the real person.
This one, sir, is the real-time data. We have just captured when Mycidac-C being doctor started, and this patient case, which is under now manuscript been written, we are in the process of publication. This, which is the very recent development in last 4 5 months. This is what I was just trying to. Meanwhile, Dr. Amit put up all this. Last year when I took over this charge, unfortunately COVID hit, and then none of the activity we could take forward because we wanted to reach out to the all oncologists and discuss what we are discussing now is a shorter, very smaller one-on-one basis. We would like to reach out to multiple oncologists and talk about it very openly, and we looking forward the the
Feedback.
suggestions for how to
So
improve in how to reach out to doctors. So this is
I
what
hope,
the new, yeah.
yeah, I hope it is visible
No
to you,
images.
Dr.
If you
Deepak.
have images, I mean, see,
Yeah,
this
images
right.
Very
definitely
small.
I'll share. So like this was actually been done in last May. So there was a case was there who, as I said, like just 65 years old male guy suffering from the squamous variety of the non-small cell lymphoma and presented with the nodules in the lung, and there was all over metastasis was there on the adrenal gland, hepatic, again the nodule was there, multiple was seen here. And this person was actually not being able to tolerate any kind of the chemotherapy here. So what doctor did, like he started with a nab-paclitaxel for this patient, and further he could not tolerate. So he need to stop this nab-paclitaxel plus uh, uh, the the therapy here. So what he did, like with the started on with the starting only, he started with a Mycidac-C here. So as I told in my session, like 0.2 ml is the starting dose, like 0.1 ml on the one deltoid region, 0.1 ml on the other region. So this was the only therapy he continued because this Brendan paclitaxel, he was not able to tolerate. Paclitaxel he was not able to tolerate. So that need to be stopped. And further, like after giving the 0.2 ml, he started with giving the another injection like 0.1 ml on the 8th day, 15th day, and further he shifted the injection to the 490 doses. And after continuing for the period of 6 months, he continued with the dose of a single injection for a month. And after doing the CT scan here, just I would like to show it to you here. This was the confirmation here.
So you will have to tell what is pre and what is post.
Yeah. So as I said, like in the case of uh, uh, pre-operative, like as I said, like nodule was there upon the left lung, and apart from that, there was a hepatic nodule was there, and there was also metastasis was found in the adrenal gland here. So after giving the Mycidac-C as a monotherapy here, all this metastasis is gone. Even the lung nodule primary was also not there. So this was found, by this oncologist here. Unfortunately, the technical error is there in my laptop.
It's not changing.
Yeah, so some technical issue is there.
Escape करके कर. कभी-कभी हो जाता है when you are on.
Now it is visible, sir.
Yeah, it is visible.
Yeah. So these all reports are there.
It is stagnant.
Dr. Amit, it will show one at a time. So you show one, remove, and then show the next one.
Okay. Just give me a second.
That's the limitation of
Zoom.
Zoom. Yeah.
True. Is this visible?
No, it will come. Yeah, now it has come.
Yes, sir. No.
So this is the first one. Let's show the another one.
Okay, no problem. You can show it later. Yeah.
You can anonymize and send it to him.
Yeah.
Yeah.
2 slides.
My question is that suppose I start using this molecule. In 2 or 3 patients I use, I see in one patient there is a good response, but I can't
Make a judgment. You can't make a judgment.
Yeah, I can't make a judgment. And second thing, if I present this in some meeting, people will again ask me that, means they will question me what you said previously, that people were questioning, they were suspicious.
Yeah,
So same thing will happen unless we have a one means experience with 1 or 2 or 3 patients will not be sufficient enough for you to promote a molecule. You understand?
Yes,
So
sir.
I think we need a bigger data set, and that can be like a multi-centric study. You can say that TMH, TMC, and other centers in the country, because then it's 1 or 2 or 3 patients. My personal experience will not be
Does
enough
not
to,
make sense.
yeah,
Does not make sense. Unless it is as a tertiary, I mean last care. Unless it is a last care where all options are exhausted,
yes,
but not as a first line or second line.
yes,
But we have to start somewhere.
yes,
So that's exactly, sir. The point is, this is what we are trying to plug in now, reaching out to doctors like you across India, and then we are now bridging this gap. How we can generate much more data. So say about 15-20 oncologists across India, if we started doing 5 patients, we will have at least 100% data.
yes,
So that is what we have created a CRF. We have created online CRF, the portal also, wherein there is no manhandling of the data point side. Our team will come, collect the database, and then you will be given the password and keys to data monitor and operator system that load the data. So from the baseline to whenever you're using. So we all that created, that CRF form being created, everything we have created now. Now, why we are reaching out to individually, we are reaching towards the understanding the needs from doctors like you, so that we can bridge those gaps. Sir, unless we try this molecule in number of patients, we won't be able to anytime justify. So these trial, which is being shown by Dr. Amit now, is concluding almost 228 patient data, which is
228
showing this kind of
patient data.
large,
That is, yeah.
large data. Sir, in my previous assignment, I invited Dr. Kapil Dhingra. Dr. Kapil Dhingra at Roche LLC, he was the man behind trastuzumab, who invented and then gone for the pivotal study in FDA for the approval. And I was shocked when he briefed me that the data presented for the approval of breast cancer trastuzumab was on 67 patient data. That's where he got approved, and then they keep on doing the trial, and then the thousands of patient data available today. So 228 patient data, I think, is a sizable data. In India, sir, whatever biosimilars, all trastuzumab biosimilar launched by all biosimilar companies, they have not gone beyond 100% data even today, but they are remaining biosimilars even today. So what my proposal would be, sir, and please correct me, and I will definitely seek your valuable advice. We'll start this program with the having CRF given to you, and then you start recruiting patient and giving them, and let's see how we generate this data going forward, which will help all of us to understand this molecule, how it behaves. Dr. Khamar, this is what we are trying to do, and then we need definitely your help also whenever we see.
Okay, so now the reason we are getting this good data is because, I mean, OS, PFS advantage is significant. So even with a smaller size, we are
Yes.
getting significant amount. So P values are significant, confidence intervals are within the range, and the HR ratio is very small. So I mean, if you look from that perspective, this is a unique Indian product, which, I mean, currently as we speak, not many people can claim up this kind of a data, which is such a small sample size. Still you can see why you need a large sample size. You need a large sample size because your response rates are smaller, OS benefit is smaller. But if you look at a gefitinib approval was based on erlotinib gefitinib approval was based on a very small number. It was not in thousands. When they did it in thousand, it failed. When they targeted, it was successful, and it was in a small number. So since we have missed that bus originally, we are reviving this because now almost all oncologists knows immunotherapy. We don't have to teach them what is the T cell and what is the active immunotherapy, passive immunotherapy, checkpoint क्या है. We don't have to teach that. That background knowledge is there. We just have to put little extra on the top of it. जैसे बोलते हैं कि दाल में तड़का लगा दो.
Yes, yes.
ठीक है ना? So I feel this is the right time to revive, and luckily we got Rajiv, and he convinced us, okay, let me revive this product. Otherwise it would have gone down the drain. Okay, because we are continuing. So as he said, we got approval for gram negative sepsis using this product last year. Okay, and it is launched and it is being used gradually. It is becoming popular. But before we launched, COVID came, so it was used in large number in COVID. Now the advantage of this product is, I mean, if I have to summarize as immunotherapy, it is active immunotherapy which enhances body's response, but it does not exaggerate immune response. So if your immune response is suppressed, it will go up, but it will not be exaggerated. So we have given this product, myself, to the tune of, see, what they are talking is 0.1 ml, 0.2 ml. I have given 5 ml IV. Okay, and still I have not seen side effects. Give 2 cc dexamethasone, give 5 cc, because it has a dual effect. It has an anti-inflammatory effect while maintaining active immunity or enhancing active immunity. It suppresses the immunosuppressive part, but it also works as a, I mean, anti-inflammatory, potent anti-inflammatory. And there is a science. If you have a time, I can go into the science part of it. How it works on the TLR, how it works intracellularly, how it works at the gene level, how it works as a signaling pathway. We have worked out all those things. Only thing is now it is a call to make it successful clinically.
Yes, sir. That's right. Sir, and this
So,
discussion
like,
with
I mean,
you
if
is
you
happening.
want, I mean, so some animal studies were done. After we did it, we transferred those to Indian Institute of Science scientist. He reproduced and he published. Same way we transferred it to National Institute of Immunology. They reproduced and published. So there are two articles on animal studies, though originally done by us, but published, done independently and published by them, and verifying that whatever we are saying is right. It takes care of immune suppression. It increases the intratumoral infiltrate of the immune cells, and those immune cells are activated immune cells. So all that has been worked out.
Absolutely, sir. And I wish to add one thing, sir. At some point in time, as Dr. Khamar was already mentioning, at some point in time, sir, we need to also come out our own invention and research and our own data. Such a robust data patient population we generate. All these companies, checkpoint inhibitors comes in India and then get away these all data, sir.
I mean,
If you start developing this kind of a data, I think you have very great paper coming in your hand, which is you yourself can get published into the and
yeah,
presented
wherever
into
you want.
various congresses happening across. So
See, I
that is
mean,
something also, you know.
see, when the immunotherapy was not a buzzword, we got oral presentation. It speaks volume for the product. Correct? Oral presentation at ASCO and World Lung Congress. How many will have with a discussant
Yes.
identified by them?
Right.
Okay. So 30-minute presentation, 20-minute discussion. So I mean, so there is a strength. We only have to prove it multiple times.
Sir, absolutely. I would quote one more thing and just borrowing from you only 2018 ESMO. I was there in the ESMO, sir. The plethora of data coming in favor of fulvestrant because now we have PARP inhibitor coming in. So there are huge
Correct.
studies coming out. You know and fulvestrant was nobody was almost using. It was gone down and have used now in combination with almost everywhere today. Fulvestrant, you see the breast cancer area. There's been multiple uses going on, but was actually gone down. So this is something, sir. When I joined, I find is really a good product, sir. And when I was reaching out to medical oncologists and talking to them, understanding with them, they identify, as you really pointed out very clearly, Dr. Deepak, that is such a wonderful data presentation now. 2 month, 3 month survival advantage OOS coming out. Why it has been missed out from the eyes of all medical oncologists, medical fraternity, India and abroad. Abroad, we didn't reach out. That's one thing. Now in the Asia Pacific area, and when we started operating now, we possibly go into Africa and other countries where we make it where the regulations are is not as stiff as the US FDA or European FDA. But India, for sure, yes, we're trying to
It's
reach
not.
out to doctors
It's
and
not.
let's see how we develop
Okay.
this
So
confidence and
Rajiv,
data.
it is not a stringent regulatory requirement. Say US FDA approved his IND, so we pass
No,
the.
in terms of number of patient, and then I'm saying that the investments
Yeah, yeah.
only
So it is basically it is more of a muscle power, not
muscle
a
power. Yeah.
science part.
Sir, I quote one more thing. My previous assignment with Zydus Cadila, and we were the only company launch adalimumab in India. So Forteo, which is the most biggest blockbuster in the world, doing almost 18 billion US dollar
20,
business,
20,
and
20.
that is not present in India. Adalimumab, Forteo. Zydus Cadila is the only company could bring out launch this product in India. And sir, what? Just because of this muscle power, this product died out, and as not, they could not go to US because the 10 US billion dollar investment upfront was there for clinical trial, and now there are at least 10 generic coming in India, Indian companies, and then prices go on haywire, and this product is got killed. And now the oral tofacitinib is available, so everything got shifted in India to tofacitinib because of the price points. So that is also one point. I mean, Indian innovation coming out, and then you are not going into the Europe or US, not getting FDA or NCC approval into any of these guidelines, and the Indian products are dies out. And then there is no other US Europe companies coming forward and then put us money. Okay, oh yes, let's co-develop this product into, and then possibly we have these kind of rights. So this is what has happened up till now. Story. But trust me, sir, if we start small steps over this product and then start identifying the suitable, of course, yes, suitable patient with the CRF in place, what we have approval from the DCGI for the doing trial on IV patients and all, we can extend even that using that product, which will definitely help these patients. And sir, very, I personally believe this, against 2.5 lakh rupees monthly per cycle cost of therapy, if we are offering real immunotherapy to patient as a less than entire cycle or less than 50,000 rupees, it is worth trying those patients who are actually deprived.
Immunotherapy for Indian patients, most of the Indian patients, it is of no use because most of the 99.99% patients don't afford.
They don't afford.
If we have a molecule which is providing equal benefit as immunotherapy, then definitely it's worth using.
Right, sir. So it is worth offering those selective patients. And of course, yes, sir, my endeavor will remain same, and I will make sure that we are organizing one program with Dr. Kumar Prabhash, wherein we are inviting approximately 20, 25, 30 eminent oncologists like you. I shall invite you soon. He has agreed upon presenting his own data. He's moderating this session, that his experience up till now with this product. And few other oncologists already are using Mycidac-C today in India. They will also share their experiences. And then therefore, we start developing the confidence building across, and then we develop this product. I think this is something we have not done, missed out, and we will be doing that. And Dr. Kumar is kind enough to come on this call and then gracing us and taking this science also once again to reach out to all of you so that we can do whatever needed, sir, for Indian patient population. That's the thinking we have. It would be really nice if you try on some of patients. My team is there in Kolkata available. We reach out to you separately, as we met those days in TMC in the evening hours when outside the corridor we met, and then I expressed the same thing. I'm still saying we will reach out to you once again and see. We'll provide you CRF, just record patient data record form, and then see how it behaves, and developing the consensus and maybe confidence.
Sure.
Any other things, sir, if I can assist you? It is really pleasure having you, sir. Thank you very much for sparing your time, almost hour and a half with us too late this evening. So kind of you, sir. Thank you very much. So Bashish and Saptarshi, please meet sir once again and see then how we can assist him.
Thank you so much, Dr. Deepak. Thank you, sir. Thank you so much, sir.
Thank you, Dr. Amit and Dr. Kumar. So nice of you being there. We all will be benefited with your expertise in this very product.
I'm not expertise. Expertise on the other side of the screen.
Thank you very much. Have a good
night, sir. Good night, everyone. Good night.
Thank you, sir. Good night.
Night, sir.
Good night.